Hello everyone. Continuing with our discussion on enzymes. Last time we discussed about cofactors. Cofactors are all substances which enhance the activity of enzyme or are essential for enzyme activity. Organic cofactors are coenzymes, inorganic cofactors are metal or non-metal ions. Coenzymes which are loosely bound to apoenzyme are called co-substrate and they leave the enzyme in an altered state like NAD. Do you guys remember this reaction from uh, glycolysis? Glyceraldehyde 3-phosphate getting converted to 1,3-bis phosphoglycerate by glyceraldehyde 3-phosphate dehydrogenase. NAD acts as a coenzyme or co-substrate that it accepts hydrogen from glyceraldehyde 3-phosphate and gets converted to NADH. So it's leaving glyceraldehyde 3-phosphate as NADH. It's then in pyruvate to lactate, pyruvate dehydrogenase, it donates that hydrogen and it is leaving pyruvate dehydrogenase as NAD. So its state gets altered, but if it is tightly bound to the enzyme, it is called as a prosthetic group. And in that situation, even if it gets altered a little bit during reaction, it returns to its original state at the end of the reaction, like succinate dehydrogenase FAD becomes FADH2, but immediately it gives those two electrons those to the electron transport chain, and FADH2 becomes FAD immediately. So prosthesis comes from the Greek word prosthethenai. It's a verb which means to add to or to fit onto. Like we have knee implants, teeth implant, which are difficult. They are also called as prosthetics, prosthesis, and they are difficult to remove from our body. But this co-substrate or coenzymes, when loosely bound to apoenzyme, can be compared to a glove, or some clothes that we wear which can easily take off, right? So functional unit of enzyme is called holoenzyme. Active enzyme or holoenzyme is protein part apoenzyme plus non-protein part cofactor. Coenzymes from non-vitamin precursors are lipoate, coenzyme Q, tetrahydrobiopterin, adenosine triphosphate, and adenosine diphosphate. So we saw that holoenzyme is made up of apoenzyme or the protein part of the enzyme and then the organic cofactor coenzyme, and there is also inorganic. So all enzymes may not have coenzyme. Some may have only coenzyme, some may have a coenzyme plus an inorganic cofactor. So this is a generalized diagram. This enzyme is like a huge molecule with a reaction happening at a cleft inside the enzyme called as the active site, where the substrate comes and binds, but the active site by itself will not be the enzyme which have coenzyme and cofactor. They will not be active till the coenzyme and the inorganic cofactor come and bind at the active site. Once they bind at the active site, the enzyme can efficiently carry out catalysis. That is what we'll see later, that the catalytic action of enzyme is because of the active site, right? So active site is a small area on the enzyme. Active site is a small area on the enzyme where substrate binds and gets converted to product. Active site is a small area on the enzyme where substrate binds and gets converted to product. It includes the substrate binding site and the catalytic site. So substrate binding site is the site which will come in the contact with the substrate initially, and then the substrate can continue to bind further with the enzyme, and then when the enzyme substrate complex is properly formed, that is when the substrate comes in contact with the catalytic site and it gets converted to product. An active site is a small cleft or crevice in the enzyme. Active site occupies only a small portion of the whole enzyme and is constituted by only a few amino acids, and the large part of primary structure of enzyme only contributes to that specific nick. You can see here it's just a diagrammatic representation. If you see the 3D models on uh, websites of enzymes, the cleft, it's like a small cleft, in a big mountain, a small cave, which is the active site of the enzyme, which is made up of few amino acids, and the rest of the primary structure of the amino acids only contributes to this specific folding and arrangement of functional groups at this active site, so that the active site becomes actually the active site, which can catalyze the reaction, which facilitates the conversion of substrate to product. Now, again, substrate binds via weak non-covalent bonds to the active site. Cofactors form a part of the catalytic site. 3D conformation of enzyme active site enzyme substrate complex. We can illustrate that by a method called as X-ray crystallography. Specificity of enzymes is due to active site. Now, specificity of enzymes could be substrate specific. That is, they could be specific to a certain substrate. They could be stereospecific, specific to a certain stereoisomer, or reaction specificity. Enzyme could be specific for a reaction on a particular substrate. We'll see one by one. Substrate specificity can be absolute, broad, relative. Relative could be group dependent, bond dependent. So absolute substrate specificity is when enzyme acts only on one substrate, like glucokinase acts on glucose. Broad specificity, hexokinase acts on all hexoses, which is 6 carbon sugars. Relative substrate specificity is specificity either on a group, functional group, like trypsin hydrolyzes peptide bonds involving basic amino acids like lysine, arginine. Pepsin hydrolyzes bonds between aromatic amino acids, phenylalanine, tyrosine, tryptophan, or bond dependent specificity, like amylase acts on alpha 1,4 glycosidic bonds, cellulase acts on beta 1,4 glycosidic bonds. Human body lacks this enzyme which breaks 1,4 beta 1,4 glycosidic bonds which are present in cellulose. That's why we cannot digest cellulose. Lipase acts on ester bonds. Stereospecificity is specific to stereoisomers. L-alpha amino acid, L-alpha amino acid oxidase converted L-alpha amino acid to keto acid and amino group, whereas D-amino acid oxidases are specific for D-alpha amino acids. I have written down these reactions for you to notice that L-alpha amino acids are the naturally occurring amino acids. They use flavin mononucleotide as a coenzyme, whereas D-amino acid oxidases use flavin adenine dinucleotide as a coenzyme. And specificity, reaction specificity. So there is one molecule like pyruvate. It can undergo a lot of reactions, and each reaction is catalyzed by a particular enzyme. Like pyruvate dehydrogenase converts it to acetyl CoA. Pyruvate carboxylase as a part of gluconeogenesis will make it oxaloacetate. Lactate dehydrogenase will make it lactate in anaerobic condition, or transaminase will make it alanine. Mechanism of enzyme action. So, how exactly do enzymes catalyze reaction? They lower the activation of energy of a reaction. What is activation energy of a reaction? Activation energy is the energy required to reach transition state beyond which reaction occurs spontaneously. Like burning of wood, like if you want to set wood on fire, you will you must have noticed that it doesn't start burning immediately. Burning of wood is an exothermic reaction, and yet it does not happen spontaneously. You need some energy, you need to invest some energy before the wood can start to burn, and that energy which you invest initially for the wood to start burning is called as activation energy. And transition state is achieved if enzymes are present. So enzymes, what they do is they just lower that activation energy. For example, there is this enzyme called as sucrase. If you try to hydrolyze that using acid, activation energy is 26,000 calories per mole of sucrose. So, but in the presence of sucrase, activation energy for breaking down of sucrose is only 9,000 kilocalorie, one third almost. So this is a graph that you can draw in the diagram, where this is the activation energy A if the enzyme was absent, and B if the enzyme is present. It's a lowered activation energy. Transition state is achieved faster in the presence of enzyme. But you can see that enzymes increase the rate of reaction, but enzymes do not alter the reaction equilibrium. That is, concentration of substrate and products at equilibrium is same with or without the enzyme. You remember that example of 17 horses that the man wanted to divide between his sons. He wanted to give half the horses to the first son, one third of the horses to the second son, one ninth of the horses to the third son. But that 17 horses division into half, one third, one ninth was not possible. So the black horse, the enzyme, made that division possible. So 18 could be divided to half. So 9 went to the first son, one third, 6 went to the second son, and one ninth, 2 went to the third son. So 17 enzymes, 17 horses got divided into 3 sons, and the one enzyme, that is the black horse, it went back. So the enzyme doesn't get affected during the reaction, and also the number of the concentration of substrates and concentration of product at equilibrium is not changed by presence of enzyme. That is, that black horse did not change the formula. First son still got half the horses, second son got one third, third son got one ninth, but that only that division was facilitated by presence of enzyme. So enzymes do not alter the concentration of substrate and products at equilibrium, which is they don't alter reaction equilibrium, but they catalyze reaction. They increase the rate of reaction by several thousand fold. Why are enzymes important? Because this activation energy of reaction you can give by increasing the temperature, by altering the pH or ion concentration. But in the body, we have to maintain fixed temperature and pH. And under these conditions of homeostasis, enzymes act as important biological catalysts where they make the reactions of our body possible. For enzymes to catalyze reaction, formation of enzyme substrate complex is essential. And this enzyme substrate complex then dissociates into product and free enzyme, as you can see in this reaction, right? This was a theory proposed by Michaelis and Menton in 1930 and is still accepted. Several theories. Now, how this enzyme substrate complex gets formed? There are three important theories for that. There is a lock and key model of Emil Fischer, induced fit model of Daniel Koshland, and substrate strain theory. Lock and key model says that enzyme का active site is a rigid pre-shaped template complementary to substrate. Substrate binds to active site of enzyme, like how a lock, how a key fits into a lock. And this theory has been proven correct for enzymes with absolute substrate specificity, like glucokinase. But it does not explain flexibility of some enzymes, especially allosteric enzymes. We'll see what that is. When we discuss regulation of enzyme activity and enzyme inhibition, we'll understand what are allosteric enzymes. Koshland's induced fit theory or hand-in-glove theory was proposed by Daniel Koshland. He postulated that enzymes are flexible and shapes of the active site can be modified by binding of the substrate. Substrate induces conformational change in the enzyme. Conformational change facilitates attainment of transition state and conversion of enzyme substrate complex to products. This theory explains allosteric regulation of enzymes. So this can be likened to hand in glove. Like you see, this enzyme's active site is not actually complementary to the substrate, but as the substrate starts binding to the enzyme, further conformational changes are taking place in the active site. And like a hand in glove, as we keep on inserting our hand into the glove starts enveloping the hand properly, and finally if the glove is a proper fit for your hand, then it will fit your hand perfectly, like a complementary the shape of the glove will become complementary to the shape of the hand. That is what is Koshland's induced fit theory. Substrate strain is when the enzyme a substrate binds to the enzyme. The substrate is strained due to conformational changes induced in the enzyme, right? This induced fit, there are conformational changes induced in the enzyme. The substrate gets strained as a result of that. As you can see here, that the active site is not exactly complementary to the substrate, but as the substrate starts to bind, there is a change in the active site of the enzyme, and then it becomes more complementary, like a glove, hand in glove. And you can also see that the substrate has bent a little. There is a strain on the substrate as a result of binding to the enzyme. And this strain facilitates the conversion of substrate to product. And currently accepted model of how enzyme substrate complex is formed and how enzymes convert a substrate to product is a combination of induced fit theory and substrate strain. Hydrolysis of glycosidic bond between N-acetylic glucosamine and N-acetylmuraminic acid on bacterial cell wall by lysozyme is an example. Now, what are the mechanism of enzyme catalysis? How do enzymes catalyze reaction? By lowering activation energy. How do they lower activation energy? There are these mechanisms: acid-base catalysis, substrate strain, covalent catalysis, entropy effects. Right? Important role of acids and bases. Mechanism that requires transfer of proton from one molecule to another. At physiological pH, histidine is most important amino acid. Protonated form of histidine functions as acid and corresponding conjugate as a base. Hydroxyl group of tyrosine, sulfatyl group of cysteine, epsilon amino group of lysine, they are all acids. Conjugates of these acids and carboxyl ions function as bases. Ribonucleose is an important example of acid-base catalysis. So you can see here that histidine at physiological pH is an important acid. Physiological pH is 7.4, right? So at 7.4 pH above 6, histidine acts as a proton donor. Anything that is a proton donor is an acid. And at pH below 6, histidine acts as a proton acceptor. So this nitrogen atom here, this nitrogen atom, that will accept proton, right? Covalent catalysis is another theory. Enzyme has a reactive group, usually a negatively charged. So covalent catalysis is enzyme contains a reactive group, usually negatively charged nucleophilic or positively charged electrophilic group, which is present at the active site of the enzyme. This group attacks a substrate that results in covalent bonding. So there is this substrate, enzyme has a nucleophilic, this electron pair is nucleophilic, attacks this carbon, and enzyme gets bound here, right, temporarily, and substrate is temporarily covalently attached to enzyme. In serine protease, covalent catalysis along with acid-base catalysis occur. Chymotrypsin, trypsin, thrombin. So, and then finally, when the product is formed, this enzyme which is bound to the substrate also gets detached. Entropy effect defined as extent of disorder in a system. Enzymes carry out decrease in entropy of reactants which enable them to come closer to the enzyme, thus increase the rate of reaction. Higher substrate concentrations, higher collision probability, hence greater reaction velocity. Even in real life, if you want to come closer to a person, you need to have time to get to know them. If there is disorder in your life, if you can't take out time to meet and talk to people, how will you come closer to them? If there is less entropy, reactants have more time to come closer to the enzyme and each other, and hence the reaction can happen. More is the concentration of the substrate, higher is the chance that they will collide with each other and increase the reaction velocity. Now, types of enzymatic reaction can be exothermic, endothermic, or isothermic. Exergonic or exothermic reaction are reactions where energy is released, reaction goes to completion. At equilibrium, substrate is about 0.5%, product is 99.5%. They are generally irreversible. Urea to ammonia, CO2 and energy by urease is a irreversible exothermic exergonic type of reaction. It's like a ball sliding down a hill. Even these reactions need a activation energy. If you see here, this reaction R is the energy of substrate, P is product. This reaction is also an exothermic or exergonic reaction, but it has an activation energy. Like burning of wood is an exothermic reaction, but you need to supply some energy, and then the reaction will start releasing energy in the form of heat as the wood burns. So exothermic reaction also have activation energy, but once that activation energy is supplied, the reaction happens spontaneously. Isothermic, negligible energy exchange, pyruvate to lactate, and it's a reversible reaction. Lactate can also be converted back to pyruvate. Endergonic reaction is like pushing a stone up the hill or pushing something uphill. Energy is actively consumed, external energy is supplied for these reactions. So kinases generally which require hydrolysis of ATP, where ATP is the coenzyme, hydrolysis releases about 7.3 kilocalorie per mole. So that energy can be used to form bond between glucose and phosphate to form glucose 6-phosphate and ADP. That's why glucose 6-phosphate is a high energy bond, because it has taken that energy of hydrolysis of ATP. Usually accompanied by coupling exergonic reaction with an endergonic reaction. Like ATP hydrolysis is an hexagonic reaction. It is coupled to endergonic reaction of glucose to glucose 6-phosphate, and that is how we bring about this reaction. So in our body, hexagonic and endergonic reactions are coupled. Like um, one reaction will form ATP, and that ATP hydrolysis will be used for fueling another reaction. So this was all for today's lecture. Next time we'll see factors affecting enzyme activity, enzyme concentration, substrate concentration, temperature, pH, product concentration, activators, physical agents, inhibitors. Thank you so much.